This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 293, recorded on August 2nd, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Michael Schmidt, is it really hot down there?
No, it's actually pretty nice. The humidity was not so bad today, and we're going to have the lower 90s today. And so I don't think it's going to be too bad. We've been having afternoon thunderstorms, which is always nice to water the grass and conserve water. And, you know, we've been one of the areas of the country that has been blessed by not having this horrid heat dome. But unfortunately, the ocean water is quite warm. The ocean is 85 degrees Fahrenheit or approximately 30 degrees C.
Wow, that's warm for the ocean.
That's scaring the dickens out of me because of hurricanes. All that warm water is energy to make hurricanes.
Well, you'll get them in September, I'm sure.
Like bowling balls, we're fearing.
All right, well, we'll do some. It's just Michael and me today. Michelle and Petra have other things to do, and apparently it's up to us to get you interested in microbiology, and we shall do that. I will start off with a snippet, which falls in the category of phage, bacteriophage therapy for bacterial infections. We've done a few episodes on this. We've also done a few on TWiV and TWivo. Michael Schmidt used to work on phage therapy, right, Michael?
Well, about 20 years ago. And we abandoned that project because the dear souls at NIH would never see fit to fund it. And the pharmaceutical industry that we had supporting us used us as their red herring. And then they went public and became a generic drug manufacturer.
Well,
but our technology was sold to Novartis and ultimately a phage biotech company. And so much of my intellectual property lives on the real world.
That's good. Well, this paper is in Nature Communications. It's called Personalized Aerosilized. That's the word that caught me, Michael. Aerosilized bacteriophage treatment of a chronic lung infection due to multi-drug resistant Pseudomonas aeruginosa. First author is Thilo Kohler. The last author is Christian van Delden. They come from Geneva University Hospitals, University of Geneva, Lausanne University Hospital, Felix Biotech, and Balgrist Hospital in Zurich. And they write immediately, you know, the clinical use of phages predates antibiotics. They were being used as soon as phages were discovered. And very early in the 20th century. In Georgia, in Tbilisi, Georgia, they founded the Phage Institute, and it's still going today. But in fact, there are some kits that the soldiers used to get in World War I, and they had little vials of phage in the kit. Very interesting.
I actually brought some of those back. I actually visited the Phage Institute in Tbilisi in 2004, and they gave me some of those vials to bring back with me. But unfortunately, they went through the X-ray machine at the airport, and it damaged the phage. When one of my colleagues tried to resurrect the phage out of the glass vial, it was unfortunately no longer alive.
Oh, too bad. No longer infectious. Yeah. Well, you know, phage therapy has continued in Eastern Europe and these former Soviet republics, but now it's seeing a resurgence everywhere else because of multi-drug resistance and pan-drug resistant bacterial strains emerging and propagating. As I said, we've talked about a number of these uses. They've phages have been used to treat surgical infections, respiratory tract infections, bone and joint infections, in one case an aortic graft infection. But there have only been a few placebo-controlled randomized clinical trials. One was for urinary tract infections, and the second was for a Pseudomonas burn wound infection. And both have showed successful treatment in a subset of patient. And Pseudomonas aeruginosa is a major target in these trials. It's a big cause of lung infections, especially in cystic fibrosis patients and patients with other diseases. And you have to treat the patients for long periods of time with antimicrobials, and hence you get multi-drug resistance. Now, the story in this paper is both scientifically remarkable and moving on a human level. As you'll see, this is a patient who was treated for a long-term Pseudomonas infection where the bacterium was multi-drug resistance. So this patient was a 40-year-old male with Cartaginer syndrome. Cartaginer, how do you pronounce it, Michael?
It's Cartaginer is the way I would do it.
This is when you don't have cilia, basically, in your respiratory tract, and you can't clear particles and bacteria and viruses like all of us would normally would. He suffered a back injury in 2014. He became tetraplegic. And by 2018, he had chronic respiratory tract colonization by Pseudomonas aeruginosa. He had monthly episodes of respiratory exacerbations that needed prolonged hospital stays. He got broad-spectrum intravenous antibiotic treatments. His isolates got more and more resistant. And then in July of 2019, he had multi-drug resistant Pseudomonas aeruginosa isolated. And there, as a consequence, he needed almost continuous hospitalization, and during which time he got antimicrobials. And he had severe bilateral lower lung lesions as a consequence. So this poor soul, he can't walk, and he's got a terrible lung infection that won't go away. He has to be in the hospital all the time getting IV antibiotics just to keep it in check. So his physicians said, well, this is a candidate for phage therapy. So they tried that in March 2020. They isolated a phage that would lyse a bacterial isolate from his lungs. It's,
from his deep lung.
His deep lung.
Yeah.
And so they tailored the phage to the bacterium. So it showed lytic activity on his last isolate. And they say the phage was resuspended in 5 milliliters of PBS and administered through a nebulizer. They sprayed it into his lungs over 20 minutes, and then over the next 5 days, he got a daily dose of 5 times 10 to the 9th PFU of this phage. And in the beginning, the first 2 days, he had an inflammatory reaction. His oxygen saturation dropped. His C-reactive protein went up. He had a febrile event. But then after that, he seemed to tolerate it better. No more side effects. His clinical condition improved rapidly. On day 5 post-treatment, he was free of respiratory obstruction, and they couldn't get any more.
Which is significant. And you know, one of the hardest things that we wrestled with phage therapy was delivery. And here, these workers have really nailed delivery, because if you think about it, if you gave it via an IV infusion, how would it get to the biofilm that is likely formed in this patient's lung? Because it's effectively on the wrong side of the lung. And so the nebulization was really, I think, one of the principal reasons that they had some level of success at clearing the infection.
I mean, it wasn't completely cleared. At day 10, you still had condensations in the lower lung, but they were stable. So on day 13, they moved him to a rehabilitation center, excuse me, and on continuous antibiotics still. Eventually, three times a week, IV therapy with antimicrobials, and he was discharged on day 56. He went home, which is quite something. And so the rest of the paper is detailing the therapy. So we'll summarize a little bit of this. So they isolated from his sputum various Pseudomonas isolates with multi-drug resistance phenotypes. The initial plan was to give him phages 5 days every month in a preventive approach, but they couldn't do that because of COVID. So
a virus stops a virus.
Yeah. So after 10 months, a control chest CT scan showed progressive clearance of his lower lung infiltrates. The right side consolidation was still there. And he relapsed on day 385. He needed more antibiotics. He got a 5-day course of phages then, 2 as an inpatient and 3 as an outpatient. That was well tolerated, good clinical benefit. In January of 2022, they tried to stop the antibiotics. He had a little relapse, got more antibiotics. This is day 868, folks. Oh, my gosh. They stopped antibiotic therapy in August 22, and in December, he had his last follow-up, and he resumed his professional activity in the summer of 2022. And I don't know what his professional activity is, but I bet it's a computer thing because he can sit in his chair and work on a computer, right?
Yes.
But it's a good outcome. We didn't get rid of everything, but it seems to be under control. So
this is really personalized medicine because they custom designed the preparation he received that was recovered from his bronchial alveolar lavage, because that's how they got the original specimen. They had to do a BAL in order to recover the microbes. And again, this lung infection was probably a biofilm, which is likely why they were unable to clear it, because at any moment in time, the biofilm is likely more quiescent than actively growing. And as you know better than me, Vincent, being a virologist, viruses need actively growing cells to do their thing.
So the combination of phage and the antibiotics, the phage are starting to loosen up some of the biofilms, and then the antimicrobials can get in there, right? But you can't get all of them, and so that's why you have some persisting consolidation. But he's able to function and doesn't need continuous antibiotics. So they have, of course, many isolates from the patient. They have bacterial isolates from the sputum, and they have phage isolates. And a couple of things that are interesting. The bacteria remain susceptible to phage throughout the course. They don't become resistant, which is really quite nice. They do isolate they make one isolate of Pseudomonas from him, and they do the whole genome sequence, and it has 53,000 single nucleotide polymorphisms in this genome, which is about 6.5 million base pair genome. Oh, man, that's a lot of changes. It also has deletions as well. And one of the reasons for that, of course, they're continuously multiplying in him, and they sustain changes which give the bacterium a hypermutator phenotype.
And it's likely that hypermutator phenotype is a consequence of the prolonged antimicrobials he's received for much of his life, because it's quite well known that Pseudomonas, if you give it some of the gyrase type antibiotics, will really mess up the genome of that organism. And more importantly, the microbe is always looking to increase its fitness, even in the presence of the continuous stress that is being faced. And the antimicrobials are, of course, stressors against the pathogen.
So basically what they see is continuous accumulation of mutations in isolates. They have multiple isolates over time, of course, and each one there are more and more genome mutations in the isolates. But these mutations, they have an effect. Some of them are responsible for antibiotic resistance, and some of them are mutators, but they don't seem to be changing the virulence of the Pseudomonas. They looked at cytotoxicity in cell cultures. They look at virulence in a wax moth model. You heard it right, a moth. Those little critters with wings that you don't want to chew up your clothing. It's a good model for Pseudomonas. It has no adaptive immune system and can measure virulence, and it's a good model for that. So they find that the Pseudomonas from this patient has not increased in virulence in the wax moth model over time. So this is the story. A good improvement after getting this patient-tailored therapy. The antibiotics weren't working on their own because, first of all, he's got this problem with not having cilia. He also has muscular issues, right? Because he's paralyzed. He can't move. And moving is a good way to help get things out of your lungs, right?
Mm-hmm.
If you're just lying down in bed all day, not good.
That's why they want you ambulating in the hospital as quickly as possible.
That's right. And of course, he can't ambulate, so he has an issue clearing things. And so what they think happened is the phage reduced the burden of his bacterial population, killed some of the bacteria in the biofilms and allowed the antibiotics to get into them. But as Michael alluded to, there are probably some bacteria in the lower airways that are not accessible to phage, and so those are going to remain. And they do say that we weren't able to obtain samples from the lower tract because it's an invasive procedure which you have to justify, and they say we didn't have the clinical justification for that later in his treatment because he had improved so much.
Yeah, I wouldn't volunteer for that procedure just for the sake of science. It's salt water on top of my lungs. It's sort of like waterboarding, except for science.
I guess you feel like you're drowning, right?
Yes, you do indeed.
They don't put you out either because you have to breathe, right?
And you have to cough at the end to clear whichever is left behind. So,
how much, what volume do they typically put in? Do you know?
That I don't recall. I sort of get creeped out by that procedure.
Anyway, one thing I didn't tell you is that they did some titration, and they clearly show that the phage is reproducing in the host. And they say this is the first patient data to show in vivo phage replication in the lung. So that's quite interesting. And of course, the key here is aerosolized treatment, right? They don't give it intravenously because that's probably not going to get at the biofilm in the bottom of the lung. But a nebulizer is the key, and that's very good. Because
it makes it, and phage are ideal because they're small already. And this will penetrate deeply into the lung if it's deposited on the right saline water particle.
Yeah. And
it will sink fairly deeply into the lung mass. And the only issue is how much EPS the Pseudomonas has produced and whether or not the phage can get through that EPS to the actively dividing cells to have their effect.
So this gentleman is 41, well, now 45 probably. Owes his life to phages, to viruses.
Yeah.
We often think of all the people who die of viral infections, but here's one that didn't because of one. And so, Michael, you think this bodes well for future treatment?
I do. And in point of fact, Intralytics, which is a biotech company in the Baltimore, Maryland area, announced on August 1st that they're going to start their EcoActive Phase 1/2A phage trial, and it's going to begin enrollment at Johns Hopkins University in Baltimore, and it's a double-blind, randomized, placebo-controlled trial to assess the safety and efficacy. Now, here it's oral administration of an EcoActive phage prep on the intestinal adherent invasive Escherichia coli, or AIEC, in patients with inactive Crohn's disease. And they're going to begin enrolling various locations within the Johns Hopkins system throughout the Baltimore metro area in Maryland. So if you're interested in that, I encourage you to go to clinicaltrials.gov to look it up, or contact the Hopkins folks to see what's going on.
So their oral administration makes sense because it's an intestinal problem.
Right. And again, it's on the outside. So it's effectively attacking the microbe where it is, as opposed to requiring the circulatory system to deliver it to the microbe.
Right.
So there may be some hope, and since this is going to be a large-scale trial, they're not custom tailoring it. So it could be interesting. Obviously, they've custom tailored it against the invasive E. coli that's specifically associated with inactive Crohn's disease. So it should be interesting. Intralytics has more of this on their website. And, if you're interested in phage therapy, I encourage you to take a peek at it.
So if 10 people have Pseudomonas aeruginosa lung infections, do they need 10 different phages to clear it?
I don't know. It really, they go to some extent to look at this, and this was again a clinical trial, but it was a one-off, and he did have a unique set of circumstances in clearing microbes and how this would behave in someone who did not have his syndrome. I don't know whether or not the microbes are different, or, it's like cystic fibrosis. The Pseudomonas in CF patients is just garden variety PaO1, which you and I can easily clear from our lungs.
It's interesting how fascinated people get about phage therapy. It's everything. If you take a non-scientist off the street, it's one of the things they ask. Hey, can you use viruses to cure infections? It's for some reason the idea that you take a virus, even though it's a phage, it doesn't infect us usually. To take a virus and cure a problem is fascinating to people. They love it. Well,
how many people read Sinclair Lewis's Aerosol, where the original concept has come from? You probably have it on your shelf, Vincent.
Michael Schmidt, I have it on my shelf. Look at that.
There it is. Aerosol.
Look at how dog-eared it is.
Yes.
The reason is that my mother used to teach this in high school English, and so it's full of her notes.
Oh, that's cool.
Her handwriting in pencil. And she's long gone, but all kinds of notes here, which she would use to teach her class. So yes, there is phage therapy in this book.
Yeah.
And here's one sentence she underlined. That summer he spent with a crew installing telephones in Montana. I love it. Yeah, it's a great, great book. So this is all about phage therapy, right?
But that goes to show you, even literary folks like Sinclair Lewis was literate in the science of his day. Yeah. And it really drives home the importance that we need to continue foster science literacy. The general public needs to know some of the basic tenets.
Yeah. Well, we're trying to do that here on TWiM. We don't have to do it alone. Other people can do it as well. All right, Michael, what do you have for us?
So, keeping on the theme of antimicrobials, I stumbled into this paper literally yesterday, and the title is called Alphagal-Bound Aptamers in Vancomycin Synergistically Reduced Staphylococcus aureus Infections in Vivo. And it's by Matthew Doherty, Claire Shaw, Leslie Woods, and Bart Weimer. and they are from the Department of Population Health and Reproduction and the California Animal Health and Food Safety Laboratories at UC Davis. And I should point out the first author is now a colleague of Michelle's at the University of Michigan's Medical School in the Department of Micro and Immuno. And it's unfortunate that Michelle is not here because she could have ran down the hall and asked him what was the most exciting day in the lab. Well, many of you know that if you write a great title, I'm likely going to take the bait and dive headfirst into your manuscript. Well, with this paper, I wasn't disappointed. First, as background for all of you who are not familiar with Staphylococcus aureus, I'm just going to call Staph aureus because there's too many syllables in Staphylococcus, is a leading cause of bacteremia in the United States with approximately 100,000 cases a year. And bacteremia meaning that the bacterium is actually growing in your blood. Additionally, this microbe is also asymptomatically carried by between 20 and 30% of the human populations. The pathogens of this genus can manifest in a variety of ways, including skin and soft tissue infections. You may see that abbreviated in the supermarket as SSTIs. Bone and joint and implant infections, and the frightening diagnosis that everyone fears in a hospital is one of a staphylococcal pneumonia, where the death rate is between 23 and 33%. And that sobering, and that coupled with the sobering statistic of how many people will develop bacteremia, they will go on to develop sepsis, and sepsis kills nearly 20,000 of the folks who acquire it from the staphylococci, which results in a death rate of approximately 17%. So in 2017, the last year that I saw statistics for, there were 119,000 people who were suffering from the bacteremia of Staph aureus, and nearly 20,000 folks died. So this is a pretty serious pathogen, and it can really wreak havoc on the patient. Now, also, unfortunately, over the previous 80 years of active use of the penicillin class of antimicrobials, for which Staph aureus was initially acutely sensitive to, we have now witnessed the widespread appearance of penicillin-resistant Staph aureus, including resistance to methicillin. And many of you have likely heard the methicillin-resistant variants abbreviation as MRSA or MRSA. And today, this species of Staphylococci carries the distinction that more than 63% of the Staph aureus isolates are resistant to at least one antibiotic. Let me say that again. For every 100 isolates of Staph aureus, 63 have at least one drug resistance trait being expressed. Moreover, many are also resistant to more than 4 antibiotics. If that doesn't frighten you as much as the global climate changes I'm witnessing, the hot water temperatures of the Charleston Harbor today being at a balmy 85, I'm terrified of hurricanes. Consider we have also witnessed the development of resistance to the antibiotics of last resort to treat infections caused by Staph aureus. The antibiotics being daptomycin and vancomycin. And as a quick review, daptomycin is a cyclic lipopeptide that exerts its bactericidal effect by disrupting multiple aspects of bacterial membrane function. And we all know that if the membrane goes kaput, everything stops. No RNA, no DNA, no protein synthesis, because you need energy. And daptomycin leads to the rapid depolarization of the membrane potential, so the cell is effectively dead by membrane depolarization. Vancomycin, on the other hand, is a glycopeptide antibiotic that exerts its bactericidal effect by inhibiting the polymerization of the molecular girdle of bacteria, namely peptidoglycan in the bacterial cell wall. And without the girdle, as the cell grows, it will literally burst from the hydrostatic pressure that is developed due to the growing cell. Now, the problem with the antibiotics of last resort, Dapto and Vanco, is they only work against gram-positive microbes. So as the title of this paper implies, the authors are going to try to do something a bit different than a straight-up approach of targeting a unique microbial process and then throw in a new compound in it or a drug. They're going to use something called an adjuvant, which means that you give something in addition to the antimicrobial. Now, this is not really new per se for any of you who have taken the branded antibiotic Augmentin. You've actually benefited from an antibiotic adjuvant combination. Augmentin is nothing more than a mixture of amoxicillin, which is a penicillin derivative, with a beta-lactamase inhibitor. Remember, beta-lactamase is an enzyme that chews up the four-member ring, the active site of the antibiotic in the penicillin classes. And clavulanic acid also contains a beta-lactam ring in its structure that then binds in an irreversible fashion to the beta-lactamase, preventing them from inactivating the beta-lactam antibiotics. And the good news about Augmentin is it has coverage for treating susceptible gram-positive and gram-negative infections. Well, even adding inhibitors or adjuvants cannot escape the rapid rate of genome evolution and the continuing emergence of antibiotic resistant strains, even against antibiotics like Augmentin. So the anti-infective community really needs to change things up a bit if they're going to make headway against our never-ending tug of war with the microbes. And today's paper will offer one such alternative strategy where here they're going to actually leverage the patient's own immune system, the patient's own pre-existing antibodies to target the offending microbe. Now, longtime listeners of the TWiX family of podcasts will recall that antibodies result from the patient's own immune system responding to a foreign antigen. And antigens can be proteins, they can be carbohydrates, can be intact viruses, they can even be intact bacteria. But here, the foreign antigen is the alpha-galactose epitope that's in the terminal disaccharide galactose alpha-1,3 galactose, which is then followed by N-acetylglucosamine, a component of peptidoglycan, in its third position, forming a trisaccharide of gal alpha-1,3 gal beta-1,4 N-acetylglucosamine, which is quite a mouthful. So hereafter, I'm just going to call it alphagal. So most importantly for the application in today's paper is that alphagal is a carbohydrate structure not produced by humans, old world monkeys, or apes, but it's commonly found across many other mammals and bacteria, especially beef. We get exposed to alphagal through our diet and the microbes in our gut. So we are likely getting continuous antigenic stimulation, which gives us chronic exposure, at least in humans, that then leads to high levels of antibodies against alphagal. And in fact, the antibodies against this molecule in humans can comprise up to 10% of our circulating antibodies and 1% of our IgG antibodies. Now, I'm going to throw this out just for fun. Can anyone say COVID-19 boosters? Think about that. If we could do that for the COVID-19, we'd really be we wouldn't need any more boosters.
Well, Michael, when the virus came out of the bat, it most certainly had alphagal on it.
It did.
But as it passed through people,
it lost it.
It lost it.
It lost it. You got it.
So the antibodies we have against alphagal, we have, as you said, we have a ton. We're no longer useful.
And in fact, that's the one bit from their introduction that's so important for everyone to read. And quoting directly from the paper, in an evolutionary context, anti-alphagal antibodies may have emerged as old world monkeys faced viruses taking advantage of the host's own alphagal surface modification, thus simultaneously losing the ability to produce alphagal epitopes and creating antibodies capable of flagging these invaders became an advantageous way to fend off viruses.
So the implication is that we lost the ability to make alphagal because it was better for us to defend against viruses.
Yes.
Right? And that's a big thing. I don't know how important alphagal was to us,
Well, I put into the show notes a paper that's on this topic, and they guesstimated that this jump occurred 28 million years ago from an evolutionary perspective. So it's in the show notes, so you guys can take a look and riff on that a little bit. In addition to the virus flagging anti-alphagal antibodies, they're also active against galactotransferase positive bacteria via the activation of the complement pathway. And we know what complement does. It's, again, a membrane perturbant. It uncouples the membrane and the cell dies. So now the authors then transition to their intent. The anti-alphagal antibodies readily react with the alphagal on the surface of viruses, bacteria, and animal tissues consumed via diet. And so many of us will now have direct experience with the approach that they're going to test.
Michael, why don't our anti-alphagal antibodies block Staph aureus?
Because it doesn't have enough specificity it doesn't have the marker. And that's the trick they're going to use. They're going to use, just jumping ahead a little bit, they're going to use an oligo that recognizes Staph aureus that they then will couple to alphagal. And so you're making a chimeric. And so then our antibodies will recognize that. Yeah. So it's effectively what they're creating is a sandwich ELISA.
Yeah, a sandwich.
And we have done everybody in America has probably done at least one COVID antigen test. So you all have experience with this. You've all done the ELISA test. You may not have known why it turned blue. The control line turned blue, but it's effectively reacting. And that's effectively what they're doing. Recall that in this.
So Michael, Staph aureus does not have alphagal, or it's just not enough, or
it's not enough. It's not enough.
Okay.
But
this aptamer, this oligo, this DNA, right? Single-stranded DNA or RNA.
Right.
This binds Staph aureus.
Yes. And that was previously established.
And what does it bind to? Do we know?
I don't know. And I didn't have, since I only found this yesterday afternoon.
Yeah, I was just reading this. I couldn't find what it's binding to.
No.
But it's binding to Staph aureus, and then they've got an alphagal stuck onto the oligo, right? Okay.
So this then flags the pathogen for binding by the approximate 10% circulating anti-alphagal antibodies
that we have,
and signals our adaptive immune system to clear the infection before mounting a new antibody response against specific pathogen-associated molecular patterns, or PAMPs. So it gets around the bottleneck of having the whole adaptive arm of the immune system, which takes time. It effectively brings in the complement straight away. So I think many of you probably have guessed now, the authors are going to test. They're going to add something that will encourage or force the anti-alphagal antibody to bind to the pathogen, irrespective of whether or not the bacterium targeted is decorated with the galactosyl transferase positive sequence. So here's where they introduce us to the aptamer, which are these single-stranded oligonucleotides that can either be a single-stranded DNA or single-stranded RNA. And the trick here is the molecules, these single-stranded oligonucleotides assume a three-dimensional conformation capable of binding small molecules, proteins, or even whole cells. And in this case, they're binding the Staph aureus. Now,
this is amazing, folks. So listen, just these DNAs fold in a way that allow them to bind to all kinds of stuff.
Yes, or it could be RNA, or it could be RNA
too. It could be RNA. Now, we know that DNAs bind proteins all the time, so that's not unprecedented.
Many of us, I shouldn't say many of us in the olden times did gel retardation assays. Do you remember those?
Yeah, of course.
And it was poly-DIC that I used.
Yeah, you add a DNA. If you run a DNA on a gel, it'll migrate a certain position. But if you add a protein that binds the DNA, it'll migrate slower.
And then you add the poly-DIC to interfere with it.
We called that a electrophoretic mobility shift assay.
Yes. Yes.
Those are the old days. Nobody does that anymore.
No one does that anymore. We have computers to do that for us now.
Nobody does experiments anymore. We just predict.
Yeah.
Well, that's not true, but it seems that's where it's going, Michael.
Yeah. However, from an anti-infective engineering perspective, aptamers have multiple advantages over antibodies, including they're smaller in size, they have low immunogenic potential, they have higher target affinity and selectivity. They're easily synthesized in vitro. You can do quality control. You can assess their purification. Good grief. Our good friend Mark Martin was recently got a bad batch of oligos that he was, fretting over checking out his PCR reactions for his students. And so he found, so you can do all the QC that normally is expected of drugs, and you can allow for modifications that change their stability and specificity in the host that they're going to try to protect. Now, previous workers in the field have already applied alphamers to control group A strep infections, the causative agent of strep throat, in vitro. And aptamers have also been used for specific binding of whole Staph aureus cells, and this was reported by cow, but they did it in vitro. And what this work is doing is actually going to demonstrate that this will work in an infection model in a mouse. So this study is building on the work of cow that found the Staph aureus aptamer, and will assess whether or not the aptamer from their study that they abbreviated SA31 coupled to alphagal could rescue, and underscore the word rescue, the alpha-1,3 galactotransferase knockout mouse from MRSA-induced sepsis, because mice have alphagal. So they wouldn't be making antibodies against alphagal because the mouse didn't have that adaptation. So they have a transgenic mouse that they knocked out the galactosyl transferase on its cells.
And they make antibodies now, right?
And they make antibodies, so they look like a human. So what the authors are going to show in this study is that treatment with both vancomycin, one of the drugs of last resort, and the alpha aptamer SA31 rescued more mice and resulted in lower bacterial loads in multiple organs than treatment with vancomycin or the aptamer SA31 alone. So it's really important to appreciate that in sepsis, the bacterial end up shutting down all of your organs. And so by finding fewer bacteria in your organs, it's very suggestive that you're clearing the infection. So collectively, these data then suggest to the authors that the aptamers or the alphagal may be an effective antibiotic adjuvant to reduce bacterial distribution and persistence during antibiotic treatment of sepsis. And right now, we really don't have any great drugs to address sepsis because it's such a fulminant occurrence and it's really hard to get a handle on it. So if you're able to add this aptamer that's going to take advantage of our pre-existing 10% immunoglobulin load, then you may be able to survive a septic insult. So let's see how all of this worked in their science of their paper. In their first set of experiments, they assessed whether the addition of the alpha SA31 would inhibit MRSA growth and cellular adhesion. What they found, and again, this is effectively asking the question, does the aptamer stick to MRSA? And what they did is they found it did indeed stick to MRSA because they saw that it failed to adhere to eukaryotic cells.
That Staph aureus failed to adhere in the presence of that. Yeah.
Failed to adhere in the presence, but it didn't interfere with the growth of the MRSA.
Mm-hmm.
So what that tells you is that the aptamer is not bactericidal or bacteriostatic. The MRSA still grow, but it did prevent it from invading our cells. And they also gave us a really nice control. They have an aptamer against flu, the flu virus in there. Additionally, it blocked adhesion to CACO2 cells by approximately 50%, a reduction that remained constant despite an increasing dosage. And when we get to the bit about how much aptamer they're giving to these mice, they're really loading up the mice. They're giving 10,000 micrograms of aptamer per kilo of mouse. So that's quite a bit of the aptamer. And it wasn't toxic, or it was still active and it wasn't being degraded by nucleases. And they have all these really elegant experiments that are effectively establishing through in vitro work the bona fides that this is likely going to work. So their approach that they're going to do is, will this aptamer likely stick to Staph aureus in the blood, thereby lighting it up for recognition by the host anti-alphagal antibody. And they also then did some stability studies in which they asked whether or not they could stabilize the aptamer by capping it, and whether or not this capping action would wake up the nucleotides and destroy, if you will, the adjuvant. And here again, the answer was no. And in fact, degradation was not observed in their nuclease experiments after 24 hours. And they found that their half-life was more on the order of 10,000 hours in water. And this is a time longer than you would likely have if you were suffering from sepsis. They then asked whether the hybrid molecule would increase phagocytosis in vitro. Now, why did they do this? Again, a brief digression into the immune system and how it addresses microbial clearance. The alternative complement pathway is this tightly regulated system, and it's the primary line of defense against infection. But many gram-positive bacteria, especially Staph aureus, are resistant to the MAC complex of the primary complement pathway, not the alternate. And so the way the gram-positives are traditionally killed by the alternative complement pathway is they're cleared via phagocytosis. And so this interaction between the host's own complement pathways and the alpha SA31 aptamer were important for them to be tested. And here again, the answer was yes. While their growth curves confirmed that you could grow MRSA in 20% serum, and it was not affected by the presence of the aptamer. Their assays measured phagocytosis showed that significantly more MRSA were phagocytized when they were treated with the aptamer as opposed to no pretreatment. So the addition of the aptamer does not appear to activate the alternative complement pathway, but it does seem to disarm the staphylococci to increase the classical complement phagocytic activity profile for this microbe. Now, again, they're trying to really optimize their aptamer, and they point out that the oligo SA31 contains known CPG motifs, and that's the repeating motif of the oligo, C, cytosine, and guanine. And this is a known motif of a PAMP, or a pathogen-associated molecular pattern. So they then next asked whether or not these PAMP motifs were impacting their phagocytosis rates. And what they did is they created a control aptamer that simply switched from CPG to CPC, thereby eliminating the PAMP motif. And again, they confirmed that this variant did indeed work. So then we get to the in vivo alphamer accumulation studies, looking for toxicity and accumulation of the aptamer in host. Now, it's again important to recall the type of mouse they used was the BALB-C knockout, GTKO knockout. So it doesn't have the galactosyl transferase. So this mouse is then similar to us in that it does not express this antigen. So the mouse routinely doesn't have antibodies. So they vaccinated the knockout mice so that it would have the antibodies. So the mice were dosed in the morning and evening with the alpha or the alphamer in doses ranging from 300 to 10,000 micrograms per kilograms per day, or 0.01 grams per kilo of mouse, or about 0.1 milligrams if you assume a mouse weighs about 10 grams. I know they're a little heavier, but I can't do math other than using in lots of 10. So, well, I mean, you get screwed up when you're trying to do it.
It's fine.
And so they then measured the serum concentration and assess each animal for adverse symptoms by giving this animal so much of this aptamer. And they saw no adverse symptoms in the mouse. So this oligo combination is likely not going to induce widespread toxicity. But when they were looking for persistence, they saw that in all mice, they saw an immediate spike in the serum level of the aptamer SA31, but then it steadily decreased over time. And what this showed me is that the antibodies in that knockout mice are finding the aptamer and they're scarfing it up.
Yep.
And they're diluting it out. So you just have to continuously dose. And the serum with the maximum dose of 10,000 mics per kg per day had significantly higher levels, but it still decreased despite daily concentration. So you're really ramping up the immune system to really make this. But that's effectively what you want when you have sepsis. You want the immune system to clear the infection. So the next experiment assessed the alpha SA31 aptamer during the MRSA sepsis model. So sepsis is an easy model to induce. You just give the mouse 1 billion colony-forming units of MRSA, and then you give it in combination with vancomycin and increasing doses of the alphamer. So all the mice that received MRSA and 10,000 micrograms per kg of the alphamer did not survive beyond 2 days. So the alphamer by itself doesn't have the ability to completely clear the infection. And that's likely due to the fact that the immune system is taking out the drug before it can find the microbe in the animal. While the septic mice, given the vancomycin at 60 megs per kg per day, plus the 10,000 mics per kg per day of the aptamer, were partially rescued. And they show us some beautiful slides showing us Kaplan-Meier survival plots, showing us the tissue profiles in the kidneys, the lungs, the heart, the liver, and the spleen. And the most remarkable ones are the ones in the lung, because here is where we really don't have any drugs for, remember, the 20 to 30% of people who die from staph aureus pneumonia. This could be a godsend for them. And their principal finding from their in vivo experiments was that the aptamer SA31 treatment plus the vanc resulted in lower MRSA organ loads. And while survival was not necessarily affected by treatment with the alpha SA31 alone, the overall bacterial load in the organs was significantly affected by the presence of the alpha SA31, especially when they added the synergy of the vancomycin. So it's really working like an adjuvant, like we saw with the clavulanic acid with the amoxicillin. That was a godsend that resurrected amoxicillin in those resistant cells, because the clavulanic acid was giving the amoxicillin enough time to find the bacterium and exert its lethal effect. So in all organs except the spleen, mice treated with the vanc plus the adjuvant, or SA31, had lower colony-forming units per meg per organs than mice treated with vanc alone. And as I alluded to earlier, this was particularly prominent in the lung, where the bacterial load of the vancomycin group was significantly higher than the aptamer vanc group. And so high loads of MRSA in the lung can, of course, result in that life-threatening pneumonia. This led the authors to offer that the reduced bacterial load in the lung seen with the vanc offers promising results for the reduction in sepsis-related issues such as pneumonia through this aptamer treatment. So in summary, what the study offered is that we can teach an old dog new tricks. taking advantage of antibodies that we already have on board, and the fact that oligos like to bind bacteria. And so if we just figure out which oligos to use, we may be able to extend the shelf life of some of the antibiotics that we already have through this interaction.
Michael, it's important that the aptamer does not bind to any human tissue on an off-target way, right?
Absolutely. And that's the beauty of alphagal and the beauty of the mouse model that they use, and the beauty of the toxicity study that they done. Because if it was having an off-target effect by itself, you would have expected to see toxicity, and they saw none.
Of course, we have to do it in people just to be sure, obviously.
Oh, absolutely. And
do you think this is going to make it into people?
That is a question for Michelle to ask Matt.
Yeah, I mean, this is based, there's no company involved here, so that's the thing.
Yes.
The company has to be interested,
Absolutely.
And they may say, well, you published it, so now we can't patent it, so it's too late, right?
Well, you can probably patent the sequence and how, the capping and the other things that they can do, because it's chemistry. Here's where the chemists get involved, and this is why we have so many different flavors of penicillin.
Well, I think it's a cool approach.
When I saw this paper yesterday afternoon, I said, okay, I was struggling finding a paper for TWiM this week, and I stumbled into this one.
You could do the same for viruses. You could get an aptamer that binds a virus and do the same thing, right?
Absolutely. So
could staph mutate to be resistant to the aptamer?
It depends on the targeting of what the aptamer is seeing in the microbe. Yeah. Now, staph has a lot of things decorating its surface. We did that paper in the early days of TWiM from Nizey's lab.
Yeah, Victor Nizey, right?
Yes, we did that in the early days. And he had that beautiful figure that Elio had on Small Things Considered, showing how the Staphylococcus was decorated. And if I can find that Small Things Considered, I'll toss that in the show notes for Ray to put up so they can see the beauty of Staph aureus.
We've been going since 2011, Michael.
Yes, and Nizey was one of the early ones.
Holy cow. He was good. I liked him.
Yeah. Yeah, he is a big shot in the staphylococcal field. He's continued to push the envelope, as they say.
Is that what his main thing is?
I think so. But, people change their projects as grants go up and down.
He's at UCSD.
San Diego.
Yeah. Cool. Good story. I like that very much because I put a link in the show notes to a blog post that I wrote back in 2009 about these antibodies against alphagal in people that can be protective against viruses that come directly from animals, right?
Yes. Well, this is the reason also, when I was doing some reading on this antibody, this is the reason we can't transplant pig kidneys into people, because they have this epitope. And so our immune system will immediately attack it, a pig organ, pig liver or a pig kidney or a pig heart. And
by the way, this is in part why we heat serum when we're doing certain virus experiments, because the serum is going to have anti-gal antibodies and from cows. Cows have
exactly a lot of it.
So you have to heat it to inactivate the complement in the serum so it doesn't lyse your virus, because complement-mediated lysis is another mechanism of the antibodies against envelope viruses. Very cool.
So you learn a lot of immunology when you dig into the background of this paper. It refreshes your old friend complement in the front part of your brain again.
All right, I have an email for you, Michael. It's from Rona, who writes, "My teeth are the bane of my existence, and I want to know everything about preventing gum and tooth disease. I perk up when Michael Schmidt carries on about caries." That's a good one.
That is.
Extra fun to have Mark on. I'm going to check out the paper and look at the photos. It was extra interesting to hear Michelle discuss the scientist. You spoke a lot about cavities in children. Many of us adults also suffer with cavities and root canals. Could you have Dr. Schmidt on an office hours and just keep the topic to dental issues? Well, Michael, you're going to be on in 2 weeks.
I think so.
Yeah,
Yes.
so you could talk about it. But she has some questions. Can they explain the science of it and how swishing coconut oil in the mouth can improve our teeth? What do you think of no fluoride toothpaste, but instead nanohydroxyapatite? Does using tamoxifen or aromatase inhibitors have the effect of making teeth more susceptible to decay since they accelerate bone loss? All right, Michael.
All right. So one of my jobs here is to introduce our dental students to evidence-based practice. And so I dropped into the show notes a meta-analysis on the effectiveness of oil pulling for improving oral health. And this is a meta-analysis. And oil pulling means you literally take something like coconut oil and you swish it in your mouth, not like we brush our teeth for about 2 minutes, but for 20 minutes.
Oh, forget it.
And that's a lot of swishing. And a lot of the data depend on the oils used and whether or not you spike the oils with essential oils. And so the bottom line from this meta-analysis is 9 randomized control trials. While the bacterial counts were reduced, there was no significant difference in the plaque index and the gingival index score between the oil pulling and control groups. And the conclusion from the meta-analysis is more rigorous and better reporting is needed in order to assess the effectiveness for future randomized control trials. I mean, I think it's 20 minutes because did you swish for 20 minutes, Vincent? I mean, I don't know.
Too much. I don't think people are going to do this on a routine basis. It's too much.
Now, for the nanohydroxyapatite, and remember, hydroxyapatite is the crystal structure that basically make up teeth. And nanohydroxyapatite is often compounded with collagen, and they compose a scaffold for bone regeneration. And I, again, did a quick and dirty lit search and found a study and data from a study from 2022 demonstrate that these nanohydroxyapatite toothpastes are either superior or equivalent to fluoride toothpaste. So we don't have fluoride alone since the ADA recommends as its standard of care that you use fluoride toothpaste. Much of the work on nanohydroxyapatite has been in the realm of reducing teeth sensitivity because the nanohydroxyapatite clogs the channels that transmit the pain, and a lot of them are using nitro compounds. But the bottom line is, again, there are very few randomized controlled trials assessing the validity of the advertisers' claims. You can find a lot of these nanohydroxyapatite toothpaste without fluoride on Amazon. But again, I look for as Daniel Griffin said, you look for the science, folks. You look for the randomized controlled trials in order to see whether or not it does indeed work.
It's pretty expensive. It's pretty expensive, this stuff.
And not cheap. And not cheap. Now, the aromatase inhibitors, again, there has been a reported fracture risk in women with breast cancer initiating aromatase inhibitor therapy, which is the tamoxifen. And aromatase inhibitors used in the treatment of breast cancer do induce bone loss of bone mineral density and have been shown to increase the risk of bone fractures in women. I don't know about the mouth. I tried to get into to see if there were any case reports out there, but unfortunately yesterday afternoon I didn't find any, and I've sent this off to my friends in Perio who routinely deal with bone loss and fractures in that population base, but haven't heard back from them. So let's hope that answers your questions, and they were all really terrific questions. And thank you, because you gave me 3 questions to ask in EBD this fall to my dental students.
Good. So we should stick with fluoride, right, Michael? That's the
We should
line.
stick with fluoride because it replaces the hydroxyl group with the fluorine that changes the dissociation constant in the presence of acid. And so you can drink those Coca-Colas and not fear that you're going to melt your teeth.
Wow, that's great. All right, that's TWiM 293. Show notes are at microbe.tv/TWiM. You can send questions or comments to twim@microbe.tv. And if you enjoy our work, we'd love to have your financial support. You can go to microbe.tv/contribute. You've got several different ways you could do that. You don't have to give a ton of money. You could give $1 or a couple of bucks a month. You know, if everybody did that, we'd be great. Why do we need money? Because we have expenses like this studio here in Manhattan. Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Hope you don't get any hurricanes in the next month.
From your lips. From your lips.
But
yes.
usually they start in August, right?
They do indeed. And, though you can always tell as the weatherman jumps up and down on the TV screen, when's it coming.
Well, good luck this year.
Thank you. Well, you got to worry too, because New York's been getting hammered of late as well.
Yeah, sometimes they make their way up here. Yeah. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.